Let's get started. Good morning, everyone. Hope you enjoyed your extra half hour in bed after the conference dinner last night. And so we have an interesting session on RNA-seq. And to begin, we're delighted to have Tomi Pastinen from McGill. Thank you very much.
Thank you. I have to disappoint you all. I won't be talking too much about RNA-seq, but I'll be talking to you about how things look different when you look at expression at single base resolution, and we're using still old-fashioned array-based technologies. I'll show you some correlations with RNA-seq though, and I think the following speaker will talk about the subject you're more interested in. But let's see if I can keep you awake at this morning hour. Okay, so my presentation is very much related to what Andy Clark presented on the first day, as well as what we heard from Joe Pickerel yesterday. So we look at allelic expression, and our intention is to find cis-regulatory variation that is heritable. We've looked at epigenetic mechanisms as well, but the heritable number of heritable variants is so large and an interesting area, so we focused on that. So I'll be talking to you about the methodology, the principles of our approaches, as well as some applications of catalogs of cis-regulatory variants, if we're able to build those. Okay, so we heard yesterday and in earlier talks that there is regulatory variation is important not only in evolution, but there's some known biomedical traits such as lactose intolerance that is caused by variation in non-coding DNA that alters gene expression in cis. And genome-wide association studies are mostly hitting non-coding variants, so a lot of people believe that some of the complex disease risk is transmitted through non-coding DNA variation. So we're looking at these non-coding variants, and specifically the ones that act locally and alter gene regulation in cis, by using a method called allelic expression. So as we heard earlier, if in an autosomal locus the two allelic copies of the transcript are equally expressed, you'll observe, or there's no cis-regulatory difference, you'll observe equal expression of the two alleles. In a lot of cases, what we actually see is that one of the two alleles is differentially expressed, and this tells you about the cis-regulatory difference, which can be either genetic or epigenetic. And one can actually use these differences that you observe in these allele ratios as a quantitative trait, and you can use them for mapping cis-regulatory variation, and I'll be talking to you about how we do that. The principal advantage of doing allelic rather than total expression. So in total expression, you measure total RNA output between individuals at different genotypic groups. And in allelic expression, you look at the relative production of the two alleles in the same cell. So these individuals, where you look at the relative production, share the same environment and transacting effects. So even if there is interfering transacting effects on regulation of the gene, you should still be able to see a cis-regulatory difference if it's present in the population. So the next presentation will be talking about how to use next generation sequencing in allelic expression analysis. So I believe that a lot of people will get much more interested in allelic expression now that you are collecting RNA-seq data, which has this intrinsic allele specificity to it, but there are some challenges to that. And we've been working many years now on an alternative approach to look at allelic expression using polymorphisms as our guide. So just as saying these single nucleotide variants in transcripts that are expressed, and then taking either single point measurements of allelic expression or averaging across a transcript. And the type of the results that I'll be talking to you today about are from this averaging across regions of the genome, allelic expression, comparing that in allele ratios in DNA and cDNA, and detecting biases in allelic transcript expression. The way we do the analysis is shown here. So we culture human cells from populations. So we look at population variation in allelic expression in order to be able to map it to local variation. So a lot of the work we've done is in the HapMap or 1000 genomes lymphoblastoid cell lines. But I'll show you some data from primary fibroblast panels of unrelated individuals, and we've worked on other cell types too. So we culture the cells, we extract RNA and DNA, and then we genotype both RNA and DNA from these panels of unrelated individuals using high-density genotyping arrays. Previously we used the 1M Duo chip. Currently we're using the 2.5M quad chip. So the 1M Duo, most of the data that you'll see today is from the 1M Duo chips. It contains an informative polymorphism every 2.3 kb on an average human gene. So once we get the allelic expression across multiple sites for each gene, we normalize the cDNA allelic ratios based on the genomic DNA allele ratios. And what we do next is we try to look at correlations of this biased allelic expression to local polymorphism. So we'll try to look at cases where a heterozygous site explains the allelic expression bias we observe in population. And I'll show you a little bit more detail on that. But before we get there, we have to clean up the data like any data. So here we see intensity, density distributions for genomic DNA and for RNA from the same individual. And as you can see, there's a large number of low intensity values from the RNA, and we have to set up an intensity threshold. And then we have to correct for differences in allele ratios, which are density dependent. We use a polynomial normalization. So we have this allele intensity dependent slope in allele ratios, which are shown here only for heterozygote individuals in genomic DNA and cDNA. But after normalization, we get rid of that bias. So what we observe now is the heterozygotes in genomic DNA on average show allele ratio of 0.5, and they do so in cDNA as well. But we observe a lot of these extreme allele ratio values at heterozygous sites, indicating of extreme allelic expression. So once we've normalized the data, what we do is we average across a number of heterozygote sites in each sample, and then we calculate this delta heterozygote ratio, which is just the difference between RNA and DNA heterozygote ratios across multiple informative polymorphisms. And the data actually behaves quite nicely. This is genome-wide allelic expression done on the 1M chip. This is from a Caucasian lymphoblastoid cell line. Biological replicates, independently cultured samples. We see relatively decent correlation. The same in different cell type. We see in osteoblast very similar correlations. Technical replicates are better. If we culture in different conditions, we see a little bit of a decrease in the replicate values as expected. So it behaves very much like any expression trait. So what we do next is shown here. Here's actually data from the Yoruban lymphoblastoid cell panel, allelic expression across this long 600 kb transcript. You can't really see much. There's about 50 individuals shown here. I'll blow it up a little bit at one portion here. So each of these black lines, there are 5 individuals shown here. Each of these black lines corresponds to equal expression of the 2 alleles, and these red ticks are all the polymorphic sites measured across the transcript that show allelic difference in the sample. So you see that this one sample shows overexpression of all the SNPs, and since all our data is phased, this effect has a direction, so the chromosome 1 from this individual always shows overexpression. For this individual, the chromosome 2, the phased chromosome 2, which are arbitrarily selected, shows underexpression. This individual shows little evidence of allelic expression, very much like this one here. So we take averages across transcripts or genomic regions and then ask the question whether any of the local polymorphisms can explain the pattern in population that we observe for this allelic expression phenotype. And here's the result from this gene. We observed that in the phased chromosomes, homozygotes are more likely for this particular SNP to show equal expression, no deviation, whereas heterozygotes carrying the overexpressed allele either in chromosome 2 or chromosome 1 are more likely to show extreme allelic expression values, and we get a linear regression-based correlation from there. So take-home messages from this is that single-point allelic expression measurements, you can see big differences between individual measurements within an individual, are noisy, therefore we need to average across multiple sites. The other feature to notice is that this individual is heterozygous at only 2 sites across a 600 kb transcript. So this is a difficult concept for a lot of people. You expect when you put 100 individuals in your test that you get 100 data points. You won't, because it depends on the heterozygosity and length of the gene, how informative each gene is. Now with the 2.5 M arrays, we're getting a little bit more informativity, but obviously there's a large number of individuals that are homozygous for long stretches of DNA and won't be informative for some genes. So we, again, using a number of SNPs helps you to get as many of the individuals informative for allelic expression as possible. So typically for a long gene like this, about 80% of the individuals are informative. Okay, so we can summarize, this is the same gene that I showed you in the earlier slide, and here's the summary of the results. So this peak SNP here was the regression graph that I showed you. So we tested allelic expression association across the whole gene region, and we observed the peak association just at the 5' end of this long 600 kb transcript. And we can look at the allelic expression ratios in only heterozygotes at this site. And here are all the allelic expression ratios collected at for heterozygotes, and we can see very consistent overexpression of A allele in all the individuals that are heterozygous here. This was actually replicated in the Caucasian lymphoblastoid cell panel, so this is a population-independent cis-regulatory variant mapping 5' to the gene. And note that most of our measurements of allelic expression actually come from the introns of the gene. So we are not looking at mRNA production, we are looking at pre-mRNA production and getting allelic expression measurements that mostly reflect transcription rates because most of our data is collected from unspliced primary transcripts. Obviously, there's not only variants that alter expression of the whole gene. Here's an example of a variant that maps to an intron of a long isoform of this gene, but the allelic expression phenotype is only observed for this alternative 5' end of the gene. The shorter and more highly expressed form of the gene does not show any evidence of allelic expression in population. So a subset of individuals express small amounts of this long isoform depending on the genotype here. So when we did the initial mapping, this was published last November in Nature Genetics, We used the HapMap lymphoblastoid cell panel, about 60 individuals, where we did population mapping genome-wide of allelic expression. We observed a couple of things. If transcript, about 50% or a little more of human genes, so population variation in allelic expression, and about 50% of those can be mapped to local common polymorphisms. And what was most remarkable to us were these very strong correlation of allelic expression phenotypes based on common SNPs. So typically more than 50% of variance in allelic expression can be explained by common polymorphisms in a locus. And we are detecting much more cis-regulatory variation than can be uncovered using standard array-based gene expression cis-EQTL mapping in the same population. And since the effect sizes are so large, these cis-regulatory SNPs, when we look at them in 3-generation pedigrees, this is data from CU lymphoblastoid family, or actually the 3-generation family, including all the children as well, where we did allelic expression throughout the 3 generations. We observed that the predicted underexpressed allele behaves in a Mendelian manner in this pedigree, and over 90% of mapped cis-regulatory SNPs show Mendelian inheritance because of the large size effects we observed. Overall, about 1/3 of the genes in this pilot study showed very strong evidence of cis-regulatory variation when we used the allelic expression mapping approach. About 75% of those associations could be confirmed by traditional methods to look at allelic expression. But one should note that the way we did this associations, where we actually didn't restrict ourselves to known annotations, we rather we made windows of the genome, consecutive SNPs, and tried to merge them into blocks of allelic expression. So we detected not only full-length transcripts that were allelically expressed, but also smaller differences such as splicing differences if we had enough SNPs in the alternatively spliced exon. So what we've been now doing is focusing. So here we have an example where an alternatively spliced exon shows strong allelic expression. So it's expressed at 4-fold higher levels if you have this SNP. So this is a single point measurement, gives a strong significance. What we've been focusing now on is actually on this type of phenomena where this isoform of this gene, the whole annotated transcript, shows differential allelic expression. Here's another good example, a 300 kb transcript showing differential allelic expression in the population. And we've been now focusing completely on full-length allelically expressed transcripts because we have the best power to accurately map those, as well as a lot of the ancillary data that is or publicly available data is useful for dissecting the associations further, such as the ENCODE data, which is not as useful for looking at post-transcriptional changes. So we're trying to focus on transcriptional changes, and we have some criteria established to detect transcriptional changes. And we do think we are quite successful in that, since most of the measurements when we look at exons and introns separately for the same genes are very consistent across exons and introns. So it looks like the effects are really affecting the primary transcripts mostly. So when we focus on differences in primary transcription, we've measured about 15,000 genes in each of these cell panels. This is African lymphoblastoid cells, Caucasian lymphoblastoid cells, and Caucasian primary fibroblast. 17 to 20% of loci shows strong evidence of transcription altering cis-regulatory SNPs. in these cell panels. And when we compare to cis-EQTLs, because we are discovering more variation, we obviously, well, we see that of the top cis-EQTLs, we see overlapping signals for about 50%. So this is starting from the cis-EQTL data, but the other way around it doesn't work, obviously, because we are discovering more. So most of our associations are not significant for cis-EQTLs. But I took Joe Pickerell's data and just looked at the direction of effect in his data compared to all our significant hits. And actually the direction of effect is highly converging even if the EQTL mapping was not significant. So if I compare sites where EQTL mapping was insignificant but we see a positive signal, or sites where both studies see a positive signal, I see very similar biases of the overexpressed allele. So if there's a 1.2-fold predicted difference based on our test, over or 80% of the alleles, so overexpression of the same allele in these two independent tests using the same cell panel. So I think we are truly seeing some more heritable cis-regulatory variation because we are removing some noise when we use allelic expression as the phenotype rather than total expression. So what we're doing now is we're using the 1,000 genomes data to catalog all potential cis-regulatory variants. I won't go into detail the way we do it, but we've established a simple scoring system to avoid effects of errors in imputation, to just catalog. We're not trying to find stronger associations. We're just trying to catalog all the potential causal variants in a region. In a region that shows allelic expression association, as shown here. So when we look at the distribution of these potentially causal cis-regulatory variants, the yellow line here corresponds to SNPs that are shared between distinct cell types, and we see this 5' peak that Joe Pickerell showed before. So most of the transcription altering variants are very close to the transcript start site, or there's a predilection there. Not most of them are not there actually. But when we looked at SNPs that are specific to a cell type, what we observe is that this curve shifts a little bit further away, and this is what we understand about cell type specific gene expression. The distal enhancer elements that are further away from the gene are more likely to be involved in that sort of regulation. So we can look at this in numbers as well. So if we overall, about 36% of cis-regulatory variants are the top associations are shared between fibroblasts and lymphoblasts. But when we look at the 5' proximal variants, so the promoter variants, nearly 50% are shared. So there's substantial sharing between cell types, but obviously there are cell type specific associations as well. Here's an example of the most typical cis-regulatory variants. So here we have allelic expression phenotypes from 3 cell panels, the fibroblast, Yoruban, and Caucasian lymphoblast. Here are the association mapping results. So the peak association is for all cell types at the 5' end of the gene. And when we look at ENCODE data, ChIP-seq data, here are activating histone marks, and we observe a peak there, obviously because it's a promoter. This is DISC1 gene associated with risk of schizophrenia. So this is the most common type of variation we observe. Here's another common type of variant we see. The gene of interest is here. It's allelically expressed. And in the lymphoblast panels, we observe a peak association, 180 kb upstream of the gene, which sits on regulatory DNA based on ChIP-seq data. But in the fibroblast, we don't have any association. So this is a distal enhancer that is cell type restricted and altered. And of course, since we're looking at two cell types and even sometimes in one cell type, we can observe multiple cis-regulatory effects for a same gene. So we're looking at cis-regulation of this gene. In lymphoblasts, we observe a 3' distal association, but in fibroblasts, we observe a 5' distal association, which are not shared between the cell types, and both of these sit on regulatory DNA as well. So when we look at our mapping results and compare to all marks of functional non-coding DNA generated by the ENCODE project, we observe very strong, highly significant enrichment of all activating or all ChIP-seq data that corresponds to active transcription. So we do believe that these independent tests are giving converging results. But for an individual site, what we need to do is we need to validate. So if we can see this global enrichment, but for individual sites, we need to validate them. So we've been using many different types of techniques, EMSA assays, chromatin, allele-specific chromatin assays, which people have scaled up recently, and we are also working on scaling up, as well as reporter gene assays. And when we look at our fine mapping in promoters and use standard reporter gene assays, we can validate majority of the results with about 10% false discovery rate. So we are slowly moving towards cataloging true cis-regulatory variants. So what we want to do with this sort of data, we obviously want to understand disease variants, for example, and here's an example of a systemic lupus-associated region from chromosome 8, which is blown up here. This is the disease association. We observe an allelic expression association here too, which causes this gene to be underexpressed, this gene to be overexpressed, and interestingly, the intergenic region is also differentially expressed, and there's no annotated transcription there. We validated the regulatory variant by reporter gene assays, and this is an interesting phenomenon that we see this non-coding or non-annotated allelic transcription often associated with allelic transcription of genes. And when we look at RNA-seq data from ENCODE, we actually observed that the BLK gene that is very highly expressed in the lymphoblast and transcribed to this direction, as seen here by the RNA-seq, there's a reverse signal or there's a signal from the reverse strand of RNA production as well. And we think that this is a bidirectional promoter that is allelically expressed. We can go into more specific hypotheses of allele-specific action. So here we actually looked at allele-specific expression of this gene in a region that is associated with asthma and type 1 diabetes, and fine mapped the expression association to this single SNP, which sits on a CTCF binding site in the ENCODE data. And we could show that multiple genes in this region are altered by the single nucleotide polymorphism that we map by allelic expression. And this is well understood. CTCF can regulate chromatin domains, so multiple genes are altered by a single variant in a CTCF binding. This is a very complicated slide, but it's just a reminder that sometimes your disease associations report of a distal cis-regulatory variant. So here's the gene of interest, Wnt4, for which we observed first an EQTL association in human bone cells. This trait was bone mineral density, so this was a relevant cell type. So we observed a disease association, EQTL association, and then we went and fine mapped this association using allelic expression, and there's a single SNP that can explain the disease association, and it sits on a cell type specific DNase 1 hypersensitive site and changes expression or changes the binding site of a well-known transcription factor. So we can land to relatively specific hypotheses of what are causal SNPs of causing disease. And this SNP, this causal SNP is actually 250 kb away from the gene it impacts. The closest gene is not the culprit here. So overall, when we look at our data against disease associations, obviously we've only looked at, in this case, I'm only showing you from 2 different cell types. So less than 15% of disease associations have a very strong cis-regulatory variant in our data. These are cases where the disease association and our cis-regulatory variant association are completely overlapping, so the top associations are shared. But when we see something, a lot of times it makes quite a bit of sense based on the known disease biology. So these are functional variants that are overlapping these disease associations, so impacting the genes shown here, which all make quite a good biological sense, and we have 284 of those in our catalog now. What's interesting when we look at these is that cis-regulatory SNPs that we observe associating with disease phenotypes look to be more tissue specific than we expect by chance. So we'll see if that holds up with the other tissues we're looking at right now. So I hope I convinced you that there's a lot of cis-regulatory variation out there. Looking at gene expression at allele specific rather than allele insensitive manner may give you some advantages in terms of finding it. And what we now need to catalog all these variants is large-scale orthogonal validation tools. A lot of the things we've been doing is very low throughput for validation. We will need validation tools. Not all of this will be statistically solvable because of the LD structure. So many linked SNPs are equally likely to be regulatory variants based on mapping. Acknowledge my group as well as all our collaborators from Canada and the States, as well as the 1000 Genomes ENCODE project and GFOS consortium. Thank you.
Time for a quick question.
So it sounds like, having thought about this problem quite a bit, you guys are actually favoring a array-based detection method, sequencing-based, and I wondered if you could talk about the considerations that inform that, and whether that's a decision that might change with time if sequencing costs drop more, or how do you see that decision?
So Vincent will tell us all about this, but from my point of view, it's more of a cost issue right now, and it won't be for long. But it's not going to be sufficient to look at mature transcripts. We have to do really deep RNA-seq to get the level of data we're getting with this fixed content microarrays. Especially we're now moving from 1M to 5M microarrays, so we're essentially having all common polymorphisms. When we do these experiments on microarrays, we can sample very low expression level genes. When you do RNA-seq, you have to do very, very deep RNA-seq, especially if you want to collect primary transcript information. So for me, it's a cost issue. Obviously, I'd love to do RNA-seq at ultimate depths because I would get the total expression level at the same time and look at in kind of real time what is the influence of transregulatory and environmental effects versus cis-regulatory effects in the same data set, which I can't do right now, except this comparing across data sets, which is not optimal.
Thanks. I think we better leave it there. Thank you.